Hello class. In previous lecture we have discussed about good laboratory practices. So let's continue with the same. So we have already discussed about good manufacturing practices, and here again we are going to discuss something more about GLP and its elements. So GLP is an FDA regulation, and it is a formal regulation that was created by FDA, United States Food and Drug Administration in 1978. Right, so it is defined as GLP embodies a set of principles that provides a framework within which laboratory studies are planned, performed, monitored, and archived and reported. So why GLP created in early 70s, FDA became aware of cases of poor laboratory practice all over the United States. So they discovered a lot um, fraudulent activities and a lot of poor lab practices. Example, some these poor lab practices were found as equipment were not calibrated to standard form, therefore giving wrong measurement, incorrect or inaccurate account of the study, inadequate test system. So these were the problems. So that's why it was started. Purpose of GLP is to certify that every step of the analysis is valid or not, assurance of the quality and integrity of the data submitted to FDA in support to the safety of the regulatory product. GLP have heavy emphasis on data recording, record and retention. Elements, as we have already discussed, that specimen sample tracking, documentation, accountability, reagent material certification, then statistical procedure, instrumentation validation, QA, certification of laboratory facilities. Yes, principles of GLP include test facility organization and personnel, quality assurance program, facility, apparatus, material, reagent, test system, test and reference standards, standard operating procedures, performance of the study, reporting, and storage and retention of records and reports. Study personal responsibility should have the knowledge of GLP principles. So what are the personal responsibility? Personals are the person who are engaged in all the activities of GLP. So they should have sufficient or complete knowledge, or they must have some skills to do the activity in a lab in a quality manner, access to the study plan and appropriate SOPs, comply with instruction of SOPs, record raw data. Study personnel are responsible for the quality of their data, exercise health precaution to minimize risk and ensure the integrity of the study. Second, quality assurance program. Access to the updated study plan and SOPs, documented verification of the compliance of the study plan to the GLP principles, inspection to determine compliance of the study with GLP principles. Three types of inspection: study-based, facility-based, process-based. So inspection of the final report for accurate results, and for description report or the inspection result to the manager and their statement. Then facilities, suitable size, construction location, adequate degree of separation of the different activities, isolation of test system and individual projects to protect from biological hazard, suitable rooms for diagnosis, treatment, control of disease, storage rooms. Then apparatus, material, reagent, apparatus of appropriate design and adequate capacity, documented inspection, cleaning, maintenance and calibration of apparatus and material, not to interfere with the test system. Chemical, reagent and solution should be labeled to indicate identity, expiry and specified storage conditions. Test system, physical and chemical test system, biological test system, record of source, data of arrival and arrival conditions of test system, proper identification of test system in their container or when removed, cleaning and sanitization of containers, pest control agent to be documented. Test and reference item, receipt, handling, sampling and storage, characterization, non-stability of the test and reference item, stability of the test item in its vehicle, experiments to determine stability in tank mixers used in field study, and samples for analytical purpose from each batch. Standard operating procedure, we have already discussed about it, that it is a written procedure for a laboratory program. They define how to carry out protocol specified activities, most often written in a chronological listing of action step. They are written to explain how the procedures are supposed to work. SOPs: routine inspection, cleaning, maintenance, testing and calibration. Action to be taken in response to equipment failure, keeping records, reporting, storage, mixing, and retrieval of data, definition of raw data, analytical methods. Performance of the study, prepare the study plan, content of the study plan, identification of the study, records, dates, reference to the test method, information concerning the sponsor and facility, conduct of the study. Reporting of the study result, information on sponsor and test facility, experimental starting and completion dates, a quality assurance program statement, description of material and test method, results, then storage, sample, reference item, raw data, final reports, etc. Then storage and retention of recorded material, the study plan, raw data samples, inspection data and master schedules, SOPs, maintenance calibration data. If any study material is disposed of before expiry, the reason to be justified and documented, index of material retained. So what good laboratory must that must contain? Area should be free from smoke, smell, dust, etc. Ensure good validation, proper illumination and proper natural light. Air condition the lab with humidity control, enough space for measuring and testing instruments. Then proper arrangement of testing, take care of all safety points including proper earthing as well as fire safety, avoid uncleanable spots on floor, wall or ceiling, establish proper area for storage of incoming sample as well as test completed samples, also provide sample collection place as well as packing and tested keeping the tested sample. Then do this for GLP, keep the things at its location after use, store heavy things at bottom and if possible on the trolleys, give name of the location to everything, follow everything has a place and everything at its place principle, prepare location list and display list, put ladder for things stored on top, identify everything with its name or purpose, follow FIFO to prevent all accumulation from chemicals. Benefit of GLP, it will give better image of company as a quality producer in global market, provide hot tips on analysis of data as well as measure uncertainty and perfect record keeping, provide guideline for doing testing and measurement in detail, provide guidelines and better control for maintenance of instruments, environment control, preservation of the test records. Conclusion, gives better image of a company as a quality producer in global market, provide hot tips on the analysis of data as well as measure certainly, uncertainly and perfect record keeping, guideline for doing testing and measurement in detail. Finally, GLP provide guidelines and better control for the maintenance of instrument, environmental preservation of test and records, etc. These are the references. Thank you very much.